Okay, hey everyone, I am Jacob. I'm a postdoc at the University of Florida in the USA. Oh, yeah, sorry. There we go. Yeah, I'm Jacob. I am a postdoc at the University of Florida in the USA. Really excited today to talk about quantifying complex insect color traits from heterogeneous images using an example with metric learning applied to iNaturalist images of dragonflies globally. So I probably don't have to remind you about the incredible diversity of visual traits in insects, ranging from color to pattern to shape. Really wide multitude of traits, wide multitude of solutions to different problems that insects have. And my research as a whole, at the broadest level, is focusing on trying to kind of disentangle what's really complex and interesting about diversity that we see. So trying to hone in on what I'm calling complex traits here. So things like color pattern. These are really important to insect performance, particularly when it comes to signaling and camouflage. I might compare this to like a simple trait in color, something like the presence of green, maybe the percent of green on the body. Whereas a more complex version of that color trait might be, what is the actual spatial distribution of green spots or stripes or what have you on the body. And as we know, that can be really important. However, most of the tools that we have are pretty designed with standardized images and manual input in mind. So these are tools like Mica, QPCA, landmarking, largely designed for smaller data sets and pretty stringently standardized images. Whereas from community science and camera traps and sources like that, these images are much less standardized, also much larger in scale. In terms of standardization, I'm talking about things like subtle variation in lighting, in the angle at which insects are imaged, in the camera sensor used, in the camera post-processing that happens. All these things can vary subtly. And in scale, of course, we have data sets with hundreds of thousands of species, maybe hundreds of thousands of images per species as well. So if we want to get traits from that kind of big data set, we might need some new approaches. So my example today is metric learning, which I'll get to what that means in a second, using this global data set of iNaturalist dragonfly images. So since I want to focus on the traits today, I'm going to cover these first few steps pretty briefly. But we start with these raw citizen science images. We feed them through grounding Dino and Sigma Anything. These broad foundational models that work pretty well on the community science images. We apply a custom UNet++ model that's trained to remove the wings specifically, because wings are pretty problematic when it comes to quantifying color. We don't really want them there. We then do, you kind of need a filtering step when it comes to INAT, because a lot of the images that go in are either not good, maybe they've cut off part of the body, there's a dead individual, problems like that. So either they're not good to begin with, or the segmentation made some kind of mistake. So our filtering step clusters these in the space of these ResNet extracted image features. So you can think of this as we're finding groupings of images, where images in each group are similar to other images in that group and separate from other groups. We can then pick and choose which cluster we want to keep or drop based on some acceptable error rate, in our case 5%, leading us to this much better data set of filtered segments. So what is phase contrastive metric learning? I call it phase contrastive. It's more broadly just metric learning. So it's a bit like a classifier, but the goal of this is actually not to classify. It's instead to figure out and quantify this complex feature or trait space when it comes to variation in the images. So we're basically finding image features, which can be various complex aspects of color and pattern and shape that provide meaningful contrast between the species. We're trying to find the ways in which species vary most visually between the images, basically. And the key there, so while the backbone of the model is basically like a classifier backbone, the key in the metric learning is this loss function, which sort of defines where our priorities lie. So what we do here is we train with, in our case, we have 353 species that have at least 20 filtered segments from iNaturalist. So we train with that data set. We then extend the inference to all around 700 species. in the iNaturalist data set. So basically when you feed a segment through the model, what you get is a 2000 length feature vector representing these contrasted differences between species. We kind of need to do some kind of dimensionality reduction. Either PCA or TSNE are both good ones, and we did PCA. And each PC kind of represents kind of the broadest axis of color and pattern and shape variation between the species in the data set, essentially. If you take a look at that visually, you might start to get a sense of what PCs 1 and 2 mean. PC1 is about 10% of the variation, PC2 about 7 or 8%. And while these are pretty small percents, I think this makes sense given just how dramatic the variation in overall dragonfly visual diversity is. You can see that species are pretty well separated. So each point here is a species. Species are pretty well separated by family, and also there's quite a bit of also providing pretty good contrast within families too here. But we really need these more detailed validation approaches to figure out what these broad axes actually mean or correspond to. A really good one is mapping existing human